they came to these conclusions. So the first conclusion is that we had abiotic synthesis. Abiotic means, biotic means to do with life. So there was no life on Earth, there was no oxygen. So they believed the first simple organic compounds were made by non-living process. Okay? So biosis means life, abiosis means the acts of life. So in this way, they synthesized the synthesis of simple compounds like amino acids or simple sugars became, these existed without any kind of cell being alive. And then these molecules, which could be amino acids, for example, they polymerized to form macromolecules. So we had abiotic polymerization of these into macromolecules, okay? So first we had abiotic synthesis of amino acids, for example, and then these may form polymers, which were the proteins, polypeptides. And then we had the emergence, means the appearance of a macromolecule that could replicate, okay? And store genetic information. So now we had like a nucleic acid, we could say. Okay, so do you understand me so far? So no oxygen, no form of life. First we had synthesis of simple organic molecules like amino acids, like nucleotides and sugars and fats, lipids, fatty acids. Then these polymerized, and then we had the appearance of genetic material. Now, what is the genetic material that is most important for us? DNA. But the scientists believe before DNA existed, there was a world of RNA. Because what do we need for DNA to be replicated in the cells or in anything? Does it automatically replicate without anything? What do we need for any chemical reaction? You could say enzyme. So without enzymes, we can't make DNA, mazboot, but there are some self-replicating RNA molecules. Okay, so they can store genetic information, number one, and they can replicate themselves, so they can make more RNA molecules. So then we had a macromolecule that could replicate and store genetic information. And then we had this living molecule, we could call it. Why do we call this kind of RNA living? Because the safat of living is being able to replicate, tikathir. So the RNA molecule can make more copies. And also, in order for the first living cells to happen, that RNA molecule stores the genetic information, it can copy itself, and then this got encapsulated. Encapsulated means surrounded by a membrane, okay? And this is what they believe formed the first living cell, which is the first primitive akthar bidaiya cell, okay? So basically, we're going to look at all of these and how the scientists believe they happened. So it's believed that simple organic molecules then formed abiotically, which means without any living organism. And these experiments started by Stanley Miller in 1953. So this is a nadariya, as we said. We cannot prove this because we can't go back millions of years. So he tested the idea that energy from lightning, lightning is bad. When you have shita, sometimes there's thunder and lightning. So the energy from lightning could have given the energy for production of these simple compounds, these organic molecules from the gases, the غازات موجود في الجلف. غلاف جوي. Okay, so we have these mixtures of gases in the atmosphere. There's no oxygen. They were mostly reduced gases like hydrogen, like ammonia, which is CH4, methane, which is CH4, ammonia, and water vapor. Okay, so this is what was present in the early atmosphere. There was no oxygen. And what he did was, he did an experiment, okay, and this is his apparatus. So in this part of the apparatus, he had methane, he had ammonia, hydrogen, and water. So this is copying the early atmosphere, what is present in the Hawa. And then in this apparatus, he had these two electrical wires that give this kind of spark. This is like the barq, like the lightning, okay? So he exposed this early atmosphere over here to simulated lightning. You know, simulate means copycat, you know, like copying the lightning. And this is the electrical discharge which will give the energy, okay, for the synthesis of these simple organic compounds from these mixtures of gases. So he had all of this set up, and then he had in this area the flask that he could test to see what has been produced in this flask. Okay, he had a sampling port over here, and he was the first person to look at this kind of abiotic production of two simple amino acids, which were alanine and glycine. Okay, so what he did, he continuously exposed these gases to the barq, you know, to the lightning, and then he checked this flask to see what has been formed. When he took a sample, he detected alanine and glycine. So this had been formed through abiotic synthesis without any cell being present. So is that clear? يعني إذا مش واضح احكي لي عشان أنا بقدر to tell you a little bit more. Okay, can you tell me what happened then? Don't be shy. Don't be shy of asking questions, okay? So what did Stanley Miller do? First of all, he had this apparatus. What did he have in this big flask of his? Which gases? Hydrogen, ammonia, methane, and water vapor, exactly. So then, what did he do? Yeah, he made fake lightning. So the spark from the two wires gave the energy. And he kept exposing, you know, the fake atmosphere like this for 2 weeks, okay? So, sorry, for 1 week. And then he took a sample from here. And what did he detect in the flask? Which compound? Amino acids like alanine and glycine. Now, these are biochemicals, we could say, Mazbut, because these are substances that are present in our cells. But this was synthesized without any kind of cell or enzymes being present. Okay, so his conclusion that some organic compounds can be produced under non-living conditions. Okay, so is that clear? He took the gases, he gave them the energy from the lightning, and after a week, he found two organic compounds that were not present before inside of this flask, which were alanine and glycine. Okay, so once he did his experiments, then other people did other experiments. So other researchers tested different atmospheres, different gases, with this kind of setup of the electrical wires giving the electrical discharge. And they found lots of different compounds, organic compounds, like other amino acids and sugars that were made, even adenine, which is a nucleotide. So they concluded that most organic molecules then can be synthesized from inorganic compounds. Okay, so we have a difference between inorganic, which are the gases that were present, and the organic, which are the compounds, the organic compounds that were produced, and they say under conditions meant to copy those of the early Earth. Okay, so now we have some organic molecules that were produced, and the energy came from lightning in both sets of experiments. So then we had models, for this process. So they believe that we cannot test exactly where our monomers for proteins or whatever came from, because we cannot go back millions of years. So they believe we still don't know the origin of DNA, monomers, and proteins. We don't know. But they may have formed in the atmosphere, in the air, or maybe they fell into the ocean. Oceans are the muhit, okay, where they got concentrated. Okay, so the organic compounds were made, they fell into the ocean, and this became called like a primordial soup, which is a shurba al-badayi, shurba in the sense that it contains biological molecules. And this was similar idea to what Miller did in his experiments. So in this way, they believe, through these processes, we had simple sugars that were made that then became complex polysaccharides, fatty acids that could form the lipid membranes, amino acids that became proteins, and nucleotides that became DNA and RNA. And then these eventually formed the first cell. Okay? And basically, other theories, they said that, we have Fathat al-Harariya al-Mayya, deep in the ocean, in the Muhit. So these are areas that are very high in temperature. Okay, so we have deep sea, Umaq al-Bahar, hydrothermal vents, Fathat al-Harariya al-Mayya, that also had some kind of role in how life could have formed on Earth. And in this area, we have a lot of chemical compounds in these vents, and we have the energy from the heat, very high temperature. So this means that there may be possible sites for formation of these organic molecules. And they believe that deep in the ocean, these vents helped the dissolved gases from the atmosphere to combine and to form the organic molecules. So some researchers believe it formed in the atmosphere. Others believe that these hydrothermal vents gave the energy from the heat that they have. And also in these vents, we have minerals like pyrites, which is FeS2. These may also have like a catalytic action, and the heat is giving the energy. So these are all like nadariya that they have. And we know that the enzymes that we have in our cells nowadays, they also contain combinations of iron and sulfur, that will be important cofactors or parts of the enzymes that are important in catalyzing biological reactions. Okay, so as a summary, we had different gases in the atmosphere. The lightning energy could have made them come together to form the monomers that we know. And that was shown by Miller's experiment. Then other people, that deep in the ocean, we have these Fathat al-Harariya al-Mayya. When I say this, I think I really know Arabic. I'm so shatara. And in there, we had the heat for the energy, and we had these compounds, like pyrite that could have helped in the catalytic reaction for those different dissolved gases to come together to form the organic molecules. So this is all what they believe. And they also believe that RNA molecules, or RNA, may be the first genetic material in the cell, we could say, because as informational molecules, the genetic material has to code for genetic information, and it has to be replicated. And we know that DNA replication in our cells needs enzymes, DNA polymerases and helicases and topoisomerases and all of those ases. So it needs enzymes to be already there. But these enzymes cannot be produced without DNA because it's the DNA that will code for the enzymes. So if there's no DNA, there's no enzymes. Okay, so that means DNA cannot be the first informational molecule because it cannot copy itself without the help of enzymes. So this is why many people now think that RNA was the original informational molecule in the cell. So they believe that RNA came before DNA as an informational molecule, number one. And this was because of the discovery of these ribozymes that were discovered in the 1980s. So these are RNA molecules that have catalytic activity, okay? And this catalytic activity is بالعكس of what the scientists believed, which is that enzymes are proteins, not RNA molecules. So this is what this is saying, that this challenges the normal idea that only proteins can act as enzymes. So once these ribozymes were discovered, this made the RNA world hypothesis more believable. Okay, so now they think that yes, RNA was the first molecule. So what they believe, long time ago, millions of years ago, we had in the environment RNA nucleotides that formed spontaneously. Now, what do we mean by spontaneously? Spontaneously means without any need for energy or information. It happens by itself, okay? So some RNA nucleotides have formed spontaneously and bonded together to form the long polymers of RNA. And then some of these RNA sequences, they believe, were more stable than others. So they didn't get damaged and they stayed and they were able to replicate as well. So the molecules of RNA that were stable and that could replicate, they survived and got more and more higher in concentration. And as the time passed, natural selection, which is التقاء الطبيعية, through natural selection, these RNA strands became more and more stable and able to replicate. So these were the most common type of RNA. And then this RNA base got enveloped into a little membrane, and this became the first cell. So before the emergence of DNA and proteins, we had cells containing RNA, we could say. So as the organisms then evolved, the DNA may have replaced the RNA as the most important Raisi store of genetic information. And the proteins then took over the catalytic functions of the cell. But RNA is still very, very important nowadays inside of our cells. So this gives like dalil that it must have been the first informational molecule. So this is what they believe, you know. Now, in order for a cell to form, we need to have a membrane around the cell, because the membrane is a very important part of the cell that will allow things to come in and things to go out and protect the cell from the environment. So if we take phospholipids, for example, okay, and we put them in water, we can produce these little automatically. We have the phospholipids coming together in this circular shape because the phospholipids, as we talked about in the last class, they have some parts which are polar and some parts which are non-polar. So hydrophobic and hydrophilic. So they always associate with each other like this. If we look at, there's no zoom here. If we look at the structure of this, we see these little vesicles, the first genetic material in the cell, we could say, because as informational molecules, you know, the genetic material has to code for genetic information and it has to be replicated. And we know that DNA replication in our cells needs enzymes, DNA polymerases and helicases and topoisomerases and all of those ases. So it needs enzymes to be already there. But these enzymes cannot be produced without DNA because it's the DNA that will code for the enzymes. So if there's no DNA, there's no enzymes. Okay, so that means DNA cannot be the first informational molecule because it cannot copy itself without the help of enzymes. So this is why many people now think that RNA was the original informational molecule in the cell. So they believe that RNA came before DNA as an informational molecule, number one. And this was because of the discovery of these ribozymes that were discovered in the 1980s. So these are RNA molecules that have catalytic activity, okay? And this catalytic activity is bil'aks of what the scientists believe, which is that enzymes are proteins, not RNA molecules. So this is what this is saying, that this challenges the normal idea that only proteins can act as enzymes. So once these ribozymes were discovered, this made the RNA world hypothesis more believable. Okay, so now they think that yes, RNA was the first molecule. So what they believe, long time ago, millions of years ago, we had in the environment RNA nucleotides that formed spontaneously. Now, what do we mean by spontaneously? Spontaneously means without any need for energy or information. It happens by itself, okay? So some RNA nucleotides have formed spontaneously and bonded together to form the long polymers of RNA. And then some of these RNA sequences, they believe, were more stable than others. So they didn't get damaged and they stayed and they were able to replicate as well. So the molecules of RNA that were stable and that could replicate, they survived and got more and more higher in concentration. And as the time passed, natural selection, which is التقاء الطبيعية, through natural selection, these RNA strands became more and more stable and able to replicate. So these were the most common type of RNA. And then this RNA base got enveloped into a little membrane, and this became the first cell. So before the emergence of DNA and proteins, we had cells containing RNA, we could say. So as the organisms then evolved, the DNA may have replaced the RNA as the most important Raisi store of genetic information. And the proteins then took over the catalytic functions of the cell. But RNA is still very, very important nowadays inside of our cells. So this gives like the deal that it must have been the first informational molecule. So this is what they believe, you know. Now, in order for a cell to form, we need to have a membrane around the cell, because the membrane is a very important part of the cell that will allow things to come in and things to go out and protect the cell from the environment. So if we take phospholipids, for example, okay, and we put them in water, we can produce these little automatically. We have the phospholipids coming together in this circular shape because the phospholipids, as we talked about in the last class, they have some parts which are polar and some parts which are non-polar. So hydrophobic and hydrophilic. So they always associate with each other like this. If we look at, there's no zoom here. If we look at the structure of this, we see these little vesicles, you know how I say that they call liposome. They call it liposome because there is no protein there. It's just made up of phospholipids. So these are... Do you have a question? Oh, well, thank you. You should tell me, not tell her. Okay, thank you. I'm so sorry, but I will ضبط الأمور, you know, but this system is really hard. I mean, I managed to get into the computer and into the system, but I couldn't get to my email, so there's nothing wrong. So you see how the polar heads are on the area where there's water, but the hydrophobic tails are not. So the scientists can take phospholipids, they can put them in liquid. As soon as they put them in liquid, we get these little liposomes formed. And if they form these liposomes in a solution that has enzymes and, you know, so we have enzymes that are the catalyst and the substrates on which the enzymes work. If we have a mixture of this stuff, the liposome can actually do simple reactions, chemical reactions that normally occur in metabolism. Okay, so the idea here that we can make, we can make these vesicles, okay, by putting phospholipids in the water, and we have the possibility that if the enzymes and the reactants are present, then we can actually form products. So these liposomes can be like active in doing some kind of biological reaction. Okay, so they believe that long time ago, we had these very simple lipids that had formed. We call them, again, primordial. So primordial means بدائية. They may have come together in the early ocean to form these liposomes. And at the same time over here in the ocean, in the Muhit, we had this catalytic RNA molecules, okay? So we had high concentration of these stable RNA molecules that could replicate and store genetic information. Then these lipids came around them, okay? So this formed, went inside a liposome that was formed, and now we have our first proto-cell or primitive cell or khalaya بدائية. Okay, so that's the whole story. Catalytic RNA molecules that had a stable sequence survived. They become higher and higher in concentration because they could replicate themselves. Then they got surrounded by the phospholipids that were present, and this formed a very simple primitive cell containing our RNA molecules. So this is what they believe. But again, it's all nadariya, okay, and we cannot prove it. Now we come to the properties we're going to look of cells and what kind of things are different between the different types of cells. So as you know, the word prokaryote. So pro means before. And karyote is talking about the karyon, which means the nucleus. So the word prokaryote means that we have a cell that doesn't really have a nucleus. Okay, it came before the nucleus came. So this is an evolutionarily earlier form. So earlier form, يعني أكثر بدائية. And if we look at the ribosomal RNA sequences, the prokaryotes can be divided into two very different groups. We have the bacteria and the archaea, exactly. And then, so if we look at this picture, we see a prokaryotic or a bacterial cell. We have this area that is the nucleoid. There is no membrane around the genetic material. Eukaryotes, on the other hand, the word eu means true, okay? And karyon means nucleus. So they have a true nucleus, which is surrounded not by one membrane, but by two. We have the nuclear envelope. So here we have the nucleus containing the genetic material, and then we have the membrane around it. And this includes all the other organisms. So we have the plants, we have the animals and the fungi, algae and protozoa, which are the single-celled, mostly, like amoeba, for example. So they all have a membrane-bound nucleus. So in this picture, we have okay, so we have the origin of life. So this is the jiddo. What do you call it, jiddo? Or the salam? Long time ago. And then here, these cells divided into 3 different parts or domains. The first domain was the bacteria. Then the second domain was the archaea. And the third domain are the eukaryotes. يا بنت وين بدك روح? Okay, so why did you come to the lecture? ليش جيت? She's not a student. Okay, this is not a good way to behave for they. So if you have any they for you, let them just finish the lecture until it's finished and then they can leave, okay? Or don't come. So these are all the eukaryotic cells on the right side, okay? So we have prokaryotes and eukaryotes, and we have 3 domains of life. What do we mean by domain then? It sounds very complex, the word domain. Domain means like groups, we could say. Okay, we have one group that are the bacteria, another group that are the archaea, and the third group that are the eukaryotes. And they want to make it sound fancy, so they call it eukarya, but that means eukaryote. So biologists then know of these 3 domains, and the more closely related 2 different organisms are, the more similarity we see in the sequence of amino acids in their proteins and the sequences of DNA, nucleotides in the DNA and RNA. So for humans, we have gorillas, gorilla, that are very similar. And if you, for example, look at the hemoglobin, I can't remember how many, but there's very, very few amino acid difference, for example, in the hemoglobin of humans and of gorillas. So the closely related organisms are the higher the similarity of the sequences in their nucleotides and like DNA and RNA, and also in their proteins. And we can say that bacteria and archaea are very, very, very different from each other. As different as humans are to bacteria, the archaea are different from bacteria. So we know, bacteria are basically single cell mostly. They don't have a membrane-bound nucleus, and we call them bacteria. We have different examples here, like E. coli, we have Pseudomonas aeruginosa, Streptococcus epidermidis. Do you know of any other names? Staphylococcus aureus, for example. Anyway, so that's for the microbiology part. And then archaea, okay, originally they were called archaebacteria because the scientists believe they're similar because they share many properties with bacteria, but at the same time they share many properties with eukaryotes too, okay? So the archaea are now actually called archaea because they're very different from bacteria. We have these archaea existing in very, very extreme environments, like the Fathat al-Harariya al-Maiya, Bahr al-Mayt, in the Dead Sea, very, very salty water. In the North Pole, where there is very, very cold environment. So we have them living in extreme environments, that are not natural. So we have some archaea that are called methanogen. So this is something to do with methane. They get their energy from hydrogen and convert CO2 to methane. So methanogen, methane, generate, to make. Halophile. Can you remember the word halo? Halo means salt. Phile means love. So these bacteria, they love salt, and they live in very salty conditions. And then we have the thermacidophile. So therm is talking about temperature, high temperature. Acido, low acidic, low pH, and phile, love. So they love high temperature, okay, very low pH, and they live in those very acidic environments like pH 2, and also in the springs, that are more than 100 degrees centigrade. So these are the hamamat ma'in, for example, where it's a little bit warm, but these are really, really hot. So it's boiling temperatures. They're living in those very abnormal or unusual environments. Okay, so we mentioned then, what's the main difference between bacteria and archaea? You tell me. Good. So when we have a nucleus, what do we mean that is a true nucleus? Because it has a membrane. So the genetic material is separated from the cytosol. And for prokaryotes, we don't have a true membrane-bound nucleus. So here we see the nucleus in the eukaryotic cell. We see the membrane, the nuclear membrane. And here we have the cytoplasm. But in the bacteria, we have the genetic material in this area called the nucleoid. The word nucleoid is like a kadab nucleus, okay? So it means like a false nucleus. And so this is in bacteria and archaea, that DNA or the chromosome is folded in this little nucleoid region, okay, which is in this similar place as the cytosol. There's nothing that is like separating it from the cytosol. And here we see that the chromosome is actually attached to the membrane, to the plasma membrane of the cell. Okay, so that's the first difference, is the membrane-bounded nucleus. Now, the second difference is the presence of internal membranes. So internal membranes are the ghishā dakhiliyya, and we know in bacteria and archaea, most of the reactions happen either in the cell cytoplasm or cytosol, or on the plasma membrane. Like photosynthesis will happen on the plasma membrane. Respiration happens in mitochondria in eukaryotes, but in prokaryotes, it happens in the cell membrane. Some bacteria do have some membrane-bound, so membrane-bound means they have some internal membranes, or sometimes they're lined with protein inside of the cell. So in this picture, we see a bacteria that does photosynthesis. It's called cyanobacteria. And here we see that the membrane is folded many, many times to form structures like the thylakoids, on which the photosynthetic pigments will be present. So they form these structures to isolate all the stuff they need to do maybe photosynthesis on here. So this is a photosynthetic bacterium with lots of internal membranes so that they can do their photosynthesis happily on those membranes. So if we have a lot of membranes, why is this good for the bacteria that does photosynthesis? They will be more surface area to be able to take all more enzymes and the stuff they need to do photosynthesis with. So that's the second difference. For eukaryotes, we have a lot of internal membranes. So eukaryotic cells, they are full of internal membranes that will compartmentalize. That means separate specific functions. We have the mitochondria, we have the lysosome, peroxisome, we have the ER, the Golgi. All of these are formed by membranes inside of the cell. So they're all membrane surrounding them. and they're separate from the cytosol. Why do you think this is good? If we have, for example, a mitochondria is specialized to make ATP, mazboot. So why is it good to have a mitochondria rather than the enzymes being present in the cytosol? To separate it,
enzymes maybe?
Yeah, so that they can concentrate the enzymes just in this mantaqa of the cell. So if we didn't have mitochondria, all the enzymes for respiration, the electron transport chain will be all over the cell, and there wouldn't be enough high concentration. But in these organelles, they have all the enzymes that they need for specific processes. So this is what we mean that they compartmentalize specific functions in eukaryotes in the organelle. So the mitochondria is specialized for aerobic respiration, for example. The lysosomes, what are they specialized for? Breaking down digestive. So breaking down lots of different macromolecules. We're going to look at each organelle and see what their functions are in the next class. And basically, we have also different types of vacuoles and vesicles, hawaiselat, naqal, that will take proteins from the ER to the Golgi to the plasma membrane. Or we may have vacuoles like the central vacuole in the plant cells that provides dagat turgia for the plant cell. And basically, each organelle has materials and molecules to carry out specific functions that organelle is specialized for. Okay, so I will just do the hudud al-niyab now, so this, and then we will finish, inshallah, if we can log in. So where do I do the hudud al-niyab?